Well, welcome to the show. Not sure what I should call you, Bob, Robert, or Doctor, or Doc.
Bob is perfect.
Okay, Bob, it is. Well, welcome to the show. I'm really excited to deep dive with you. Now we have back to back, so I think we're going to focus more on your backstory on this first one, and then we're going to dive deep into testing results and what you've accomplished over the last number of years next week. So,
Right.
so let's start with your background, my friend. Like, where'd you come from with your education, and how did you end up here today?
Well, I could be Steve Martin and say I was born a poor black boy in Brooklyn, but I think I'll forgo that. Okay. I was born in New York City, of all places. My parents moved out to Long Island, and I grew up in a very nice neighborhood called Great Neck, New York on Long Island. Got my education there. Then went on to get a bachelor's degree at the State University at Buffalo in biology, where I gained my first experience in mycology, the study of fungi. Okay, my advisor was a very famous mycologist at that school, and I really glommed on to what he was doing. He took me under his wing. Then I graduated in 3 years and went on to a double doctorate in Syracuse, one in ecology from Syracuse University and one in plant pathology from the SUNY College of Environmental Science and Forestry, which is located on the Syracuse University campus, even though there are two separate colleges. Finished up there too quickly, as far as I'm concerned. I had a double doctorate the week before I turned 24. People looked at me and said, "You understand no one's going to hire you, okay, in a professorial job at the age of 24. And I said, "Yeah, I can understand that." So I was very fortunate to be accepted into a postdoctoral fellowship at Cornell University, right down the block. Okay, I stayed in upstate New York for my entire education, majoring and studying phytopathology. And the specific thing that I worked on for 2 years, which I'm glad I did for the experience I got, was studying late blight of potato. Okay, as I remind people, if you're Irish or of an Irish ancestry, you should know everything about late blight of potato, because there's about a 98% chance that's why you're here. The famous late blight epidemic of 1849 literally killed 4 million people from starvation in Ireland, and many of them emigrated to the United States, where we got our Irish population from. At that point in time, I really began looking at soil microorganisms. That fascinated me. The biology, the biological activity of soil-borne organisms. For example, late blight, which is a nasty group of organisms called phycomycetes, overwinter in the soil. Okay, that's how they survive. Places like upstate New York and Ireland are cold places. You would think that a foliar disease would die, and it doesn't. It creates chlamydospores, it goes into the soil, it interacts with other organisms, and I began becoming really interested in that at that time. Well, after 2 years, I left. Cornell was ranked the #1 plant pathology institution in the country. The #2 ranked institution was North Carolina State University in Raleigh. Huge plant pathology department. When I arrived there were 40 faculty members in plant pathology studying everything. And I was fortunate enough to get that job and began that job. And to draw out the story to the end, I spent 37 years at NC State, working my way up to the rank of distinguished professor. and worked on many, many different topic areas. But the most important topic area, which I want to focus on today, is that of soil microbiology. When I first arrived there, they assigned me to a horrible problem. Okay, of all things, North Carolina, up in our mountains, is number 2 after the state of Oregon in producing Christmas trees, of all things. Okay, the industry today is worth over $3 billion a year. Well, when I arrived, it was amazing. 2 days after I arrived on campus, they drove me up to the mountains to show me these huge plantations, sometimes 1,000 acres or more, of Fraser fir. That's our native species to the mountains of North Carolina, which are the highest in the eastern United States. And I said, oh my goodness, why are half the trees brown? And they said, welcome to our epidemic. And what it was an epidemic of a fungus called Phytophthora cinnamomum, the same group of Phytophthora infestans, late blight, except cinnamomum is strictly a soil-borne pathogen. And it literally was wiping out more than a third of the Christmas tree crop across the mountains of North Carolina. Again, a soil-borne pathogen. That's a lot of money, considering it takes 10 years to grow a Christmas tree. So, if it knocks it out in the first year, you could do something, you could replant it. On the other hand, if after 9 years, all of a sudden you have a brown Christmas tree, on that tree, you just lost your shirt. So it was a big problem. And I took it up, got a bunch of graduate students, and indeed was able to find a lot about the biology of this fungus, of this tree, certain genetic interactions, certain microbial interactions. Okay, and we tried all kinds of technologies, from natural compounds all the way up to chemicals, okay, to see what we could do about it. And to make a long story short, because it continues today, research still goes on. It is nowhere near the problem that it was 40 years ago. But what the way they're dealing with it now, which is an outstanding program, is they're doing a bunch of breeding, trying to use genetics to select individuals that are resistant and being able to then breed those trees into making a robust industry. But I left that behind fairly quickly and got involved in all sorts of soil microbial problems, including nematodes. I love talking about nematodes because unless you're a farmer, okay, I would honestly say that 98% of even the educated American public doesn't know what a nematode is. Yet, on a national and global basis, okay, nematode infestations, we'll simply leave it as being microscopic roundworms. These are microscopic roundworms. There are good ones and there are bad ones. But the bad ones, according to the UN Food and Agriculture, FAO, which keeps track internationally, nematodes alone, okay, are the cause of over $150 billion in crop losses every year. And one of the fascinating things about nematodes is up to very recently, and we could talk about that next week, but up until very recently, a farmer had to use the most horrible, toxic poisons known to man. And I've got to tell a story right now because it is something that you want to talk about a screw up. I'm at Cornell, and we have hundreds of acres of test plots doing all kinds of work, okay, to try to look at potatoes, which is a major crop in the state of New York. And we had nematodes, horrible nematode problems. So in the spring, when they would disc up the fields and put in the plots, they would put down, of all things, furidan, which is now banned. Furidan was an incredibly toxic compound, an organophosphate, similar to that of Temic, which is now banned. If I took my hand out and put a dot of Temic, pure Temic, in the middle of my hand and rubbed it in, within 30 minutes, I would go into respiratory collapse and die. That's how potent these things were. But I'll be damned, they killed nematodes. Okay? Well, our field manager we decided to put Furidan in our plots. And the way Furidan was applied is that the powder was put on little corn pieces. And they would go up and down the road with a tractor and lay down the corn pieces, and then behind it, they'd cover it up in the row. Well, I'll make a long story short. They did that one of the years that I was there. And next morning, I was up bright and early out to the field to start working on the next phase of our project. And I come out to the field and I drive up and I go, what the? There were probably 1,500 dead birds lying in the field, as in dead. Crows, sparrows, starlings, red-winged blackbirds. Apparently, the guy did not cover up those corn pieces. The birds swooped on down, said, wow, free lunch. They ate a piece, and within a matter of minutes, they died. Okay. That really set me off. I'm a young professor at this time, and I really, really was perturbed, not simply from the ethical standpoint of what we were doing, but then it came around as to what are we doing to human beings? If this material is in the soil, obviously some of it or its byproducts are going to be taken up by root systems. Okay. And I'm very young. This was back when chemicals were the thing, guys. Okay. It's still 1979. I want you to get that date set. Okay. This is long before people started talking about organic agriculture. So it really made an impression on me. And I decided at that time that I was going to do everything that I could possibly do to find, in quotes, natural solutions to not only insects, diseases, nematodes, but also how can I make a plant grow faster, better, stronger by using, in my case, microorganisms, microbes that would do all of those things. Well, back in the 60s is when there were certain laboratory experiments done with microorganisms. One of the first ones that was discovered in the 60s was Bacillus subtilis, where people were doing experiments. The other one is a fungus called trichoderma, a green fungus. And there were some esoteric papers that were popping out, basically describing, geez, we put this here, we put this there, and the problem went away. And I'm saying to myself, well, according to, again, scientists that are smarter than I, when it comes to the soil, there's probably no less than 50 million different microorganisms living in that soil. I'm not talking about numbers, I'm talking about species throughout the world, okay, in which probably 1% of them have been identified, and 0.1% of them have actually been screened for biological activity. Where was most of that work done? Let's go back to our good friend Alexander Fleming, okay? He discovered penicillin, a bread mold, okay? And lo and behold, research started on antibiotics. And if you take a look at all of the numerous hundreds of antibiotics that are on the market today, no less than 40% of them are natural products, and no less than 60% of the 40% come from soil. Living organisms, bacteria, fungi, actinomycetes living in soil. Okay? So again, I'm a plant doctor, not a human doctor. And I say to myself, okay, we are losing hundreds of millions of dollars, billions of dollars in crop losses. We're having poor reactions from fertilizers. Our farmers have to have insurance because of all the vagaries of nature, okay, that prevent them from being able to get a crop. Can we look to the natural world in order to find organisms that produce, and here's a key word, secondary metabolites? In other words, our organisms, okay, we'll talk about our products, but our organisms live on carbon and water. That's it. That's what they feed on. Our bacteria, our fungi, our actinomycetes, our rhizobacters, okay, ectomycorrhizae, endomycorrhizae, their major food source is carbon and water. Well, soil that has organic matter has carbon. Okay. Without water, well, you got a problem. Okay. We won't discuss that. Okay. Obviously, if you're dealing with severe droughts or incredibly arid environments, okay, water is a limiting factor. But interestingly, you can isolate microorganisms from the Gobi Desert and the Sahara and our western deserts in the United States that might receive an inch of rain per year. Okay, Mother Nature is incredible in terms of doing this. So that leads into the next story. What I did over 4 decades and still do today is, over my academic career, for all kinds of reasons, whether it was going to a scientific meeting, whether it was to do cooperative research with colleagues in the Amazon or in Germany, I worked for a while, I used to bring back soil samples legally. I got a whole bunch of APHIS permits to be able to import these soil samples back to my lab, under the guise that when I was done with them, I would have them autoclaved, incinerated. That's important. Okay, you don't want to be spreading pathogens or new insects around. And I'm very cognizant of that. And I did a lot of work with APHIS. APHIS, by the way, is the American Plant Health Inspection Service. Okay, that's where if you go to an airport, okay, the big signs, you're coming in from South America, it says, do you have any plants, soil, or food? Go to the red line. And of course, most people don't go to the red line. But the idea is that they have inspectors there that are going to go through your bags to make sure you're not bringing something in that's going to endanger, in our case, the United States. Canada has very similar programs. Europe has draconian programs about bringing foreign material in. I had my permits, everything's cool. So what happened? Over a 40-year period, I was collecting isolates from all different types of environments. And I was particularly interested in what's known as extremophiles. Those are organisms that live in extreme conditions. For example, if I go up to Fairbanks, Alaska, and go into their boreal forests and start digging up soil, I am going to find organisms that live basically frozen in tundra year round. You know, not anymore, but we won't get into climate change, okay? But basically, you can isolate those organisms. I could go climbing a 14,000 foot peak in Colorado, okay, or go into the lower forests of Mount Rainier, okay, where the forests, the Krumholtz dwarf forests are there, dig up soil samples. And now I have alpine organisms that have evolved over millions of years to survive in those environments. And by the way, they're there for a reason. Okay. They live in conjunction with the natural plants that are there, which is where we'll get to in a while. So I now have a library of organisms. I bring back these soils. I obviously isolate from them. I standardly use 7 different selective antibiotic agar in order to be able to pull out different organisms. And I might be looking for a single dot on an agar plate, and I'll take a pin and I'll touch it and I'll transfer it. And lo and behold, it's something new, okay, that I haven't seen before. And then I have to go and isolate it and identify it. Are there questions?
Yeah. So we
Yes.
got a lot to unpack, and I'm sorry to interrupt you because I
No,
don't. I like to let people spool out so that you're not
great.
losing your train of thought and you're getting what you want to get across. But
I'm good.
yeah, so I want to reel it all the way back in. So you studied mycology. Was it specific to mycorrhizae or saprophytic?
Very interesting. The first things that I studied were ectomycorrhiza
Okay.
on pinus. Okay, pine, spruce, fir. Okay, that's where I began my first studies. Published several papers on it. In fact, okay, but all that did was stimulate me to go further. You know, one of the beautiful things about being an academic is in the old days we had a thing called academic freedom. That's gone today. Okay, now you do what you're told, or you get fired. I don't want to get into that. But basically, so long as I could fund my laboratory, fund my grad students and postdocs, I was free to work on anything I wanted to work on. And I guess people call me a little bit ADD, which you've probably figured out by now, okay? Because I like being all over the place. And over my career, I worked on dozens of products that resulted in over 300 peer-reviewed publications. Okay, you take a look at my track of publications, you'd say this guy's a psychopath. Okay, he's publishing on this, he's jumping to that. He's now doing tropical ecology. You know, what's going on? Okay, but that was the freedom that I had. But it was that initial professor, Dr. James Haynes, Buffalo, okay, who first got me motivated into looking at the fungi. And it went on from there, obviously, into dealing with soil, bacteria, and other organisms.
So just for the audience sake, so they understand, that type of fungi is like a glove. It
Yes.
fits over the root. It's not,
Yeah.
it's not the one that goes into the plant veins. It's not what we call the endomycorrhizae. So
Correct.
it's very unique. It is a very interesting organism in the result that it has a mutual relationship, but it also harvests nutrients from the direct vicinity in which it's located.
That's correct.
Right.
The way I put it, when I describe it to people, is here's a fine root on a plant. Okay. And the fine root has a certain surface area. It has cells, it has root hairs and everything. In the right environment, when an ectomycorrhiza sees that root, it puts this giant sheath over that entire root, sometimes increasing the surface area by 20x, 20 greater amount of surface, which not only allows for absorption of water and nutrients, but as you just stated, and boy, there's some great papers been recently published, the chemical effects of the symbiosis between the root and the fungus is astounding. Okay, the root is feeding the fungus. The fungus is producing hormones, enzymes, antibiotics that protect the root and actually make it grow more robustly. Then
it's those secondary and tertiary metabolites.
the metabolites are,
Yep.
it comes down to a very simple thing, and you don't want to simplify things too much. But the living world is just a sack of water and biochemistry. That's it.
Yep.
It's all chemistry. Okay.
Yep.
And the amount of things we need to learn about it is astounding. But since you're on the topic, okay, let's talk about endomycorrhizal, because I've done a huge amount of work with that too. Here we have our root, okay, and endomycorrhizae, which are totally different species, rhizobiums and things that are found in the soil, or you can inoculate the soil, which is what we do with endomycorrhizae. They don't go around the root, they actually go inside it. And for more than 40 years, plant pathologists were convinced that endomycorrhizae were pathogens because they were penetrating the root, they were going inside, they were putting their hyphae, their little hairs all over the place. You could stain them, okay, with saffron in, look under the microscope, and you see all this red stuff in the root and everything. And it wasn't until much later on that they said, holy cow, not only is it not a pathogen, but now look at the physiology of the root and the plant that have these organisms, soil organisms in them, and the benefits that they show to the system.
I believe it was Chappie that
Yes,
was the one that brought forth that information.
that is correct. Fairly recent in the scheme of
I think it was the 70s or maybe the 80s.
It was. As I said, before then, people was, oh my God, we have this pathogen that's invading our roots, when in fact it was no such thing.
I'm very blessed in that I've had a dear friend of mine, Dr. Everin Chavez, who worked side by side under Chappie. Basically, the way you were inspired, he was inspired.
Right.
And he's gone on to do some incredible work.
Yep.
In that field, and also in truffles and
different
and that are both, you know,
that's one of my dreams. When I retire at 95, I'm starting a truffle farm.
I knew I was going to like you.
I got to make some bucks here somewhere.
Yeah. All right, so the farmers in France right now are suffering horrific losses in the truffle.
Yes. Yes.
So there you go. There's where you're going when you get to 95. All right.
That's
but you solved the tree problem. Well, now it's time to go truffling.
The truffle hunting. I haven't decided on pigs or dogs yet. So we'll
all right. So we're just scratching the surface on so many chapters that we can talk about.
Yep.
But I'd like to just kind of keep scratching on nematodes. So I'm sure you're very aware of Russ Ingham.
Yes, I am.
He would tell me that, and this was sad. I was dear friends with Dr. Lane. She was my inspiration, right?
Yes.
She took me under her wing and just showed me the whole world of what we're talking about.
Right.
And Russ would have to come out certain times of the year for a conference or whatever to the East Coast because they lived in Oregon. And she was at Rodale, so he'd show up at Rodale, and she'd be like, "Oh, damn it, Leighton, can you babysit Russ this afternoon?" And that's what she would tell me. And
right.
Hell yeah, are you kidding me? I'd be more than happy. Well, what does he want to do? And she's like, "Well, just figure it out. Just get him out of my hair until I finish today, and then I'll deal with him when I'm done." So I'd grab Russ and I'd be, "Hey, let's go down to the pub. They have wonderful food. They've got ice cold beer, whatever you want.
Right.
And we'll hang out." And so I'd get to sit with a table with him for 4 hours getting into this guy's head. And it was just like the most amazing experience, because at that point I was wise enough to know the right questions to ask.
Yes.
And it would excite him because he had somebody that was stimulating all the knowledge that he had bubbling up in him about all of these different fascinating aspects of the nematodes. And like you said earlier, we only know, we've identified 1%. That's bullshit. They're coming up with new species every day. We probably haven't even understood a fraction of a 1%.
Right.
And studied them? No, not even close. But that wasn't the point. The point was that he would tell me these stories of, his work and how he had to be a pirate. Like he had to literally sneak around because if they found out that he was studying beneficial nematodes, they'd have crucified him, right? So he and Lane would be like, Lane would be like, you're killing good ones, and he's like, there's no good ones, and he was telling me this, and I'm like, what do you mean there's no good ones? I mean, of course, there's always got to be the positive, the yin or the yang to everything.
There are very good ones. Yeah.
Right. And so she basically explained to him, like, honey, like there are so many more beneficial than there are bad ones.
Pathogenic, correct. Yep.
Way more beneficial ones.
Correct.
And so you've got to back off and start looking at ways to identify or dissect or figure out a way to snipe the bad ones without killing the good ones.
Yes.
Because if you lose the good ones, they're going to outcompete. The bad ones are going to outcompete them all.
Sure.
So oh my God, we would have such fun. And some of the most fascinating stories he'd tell me was like this one nematode that was so smart. It would whip its tail around the leg of an insect.
Oh, yes.
Fly itself up into a tree and then attack that tree.
Entomophagic nematodes. They're fantastic. And even more fascinating, what about the not the entomophagic, the nematophagic fungi that actually choke the nematodes to death?
Oh, yeah, the circle ones.
Yeah. I mean, I've seen that under the microscope, and I'm jumping up and down.
Oh,
there are all kinds of natural fungi, good fungi in the soil, that when they come across a pathogenic nematode, they lasso it like a cowboy and they choke it to death. I mean, who would have thought of such a thing, right?
Dude, I've seen it under the microscope too, and it's the craziest thing. And it's funny because that fungi is kind of like what we would consider facultative.
Yes.
It can live in the anaerobic or the aerobic environment.
Correct.
But it really seems to proliferate in the pathogenic.
Here is something to think about, and we'll talk about it later. There is a common thread that's going to run through everything we talk about. And the way people talk about big pharma is no different than talking about big ag. Okay? When it comes to finding funding, okay, if you're going to do research, you need money. You have to have some. Okay, otherwise you can't go and do what you're going to do and run your lab. Basically, one thing I'm going to be very careful about in my discussions here, I'm not going to use names because they're so obvious that I don't even want to say them. Okay, I don't want to use names of competitive products to our product because I have been through the ringer in trying to vet their materials, and the discussion that I would have is vitriolic. So I'm just not doing that. But here's the bottom line. Big ag. Okay, by the way, my lab is in the Research Triangle Park. That is the home of every major ag chemical company in the world. Period. Okay. There's 74,000 acres that are covered by ag chemistry laboratories. Germany, Switzerland, China, and of course, all the American corporations. And if you want to find these corporations, okay, just go to the Fortune 100. They're all there, okay? These are multi-billion-dollar, multinational corporations. They make chemicals. Lots of chemicals. And they come out with new ones every year. To try to break through that veil, and of course, one of the jokes is that one of the labs that's 10 miles south of where I actually live, the joke is that they have 5,000 employees there, and the employees have more attorneys and MBAs than scientists. Truth. True fact. Okay? So the bottom line is that up until now, and we are in the most exciting phase of what we'll talk about right now, today. Okay? Finally, enough people are raising hell about poisoning our food, our soil, our environments, our ecosystems, combined with the fact, which they don't want to talk about, but one of the jokes that I tell all the time, okay, I had a 40-year academic career. I produced a hell of a lot of PhDs. Where do you think most of those PhDs work? Okay, at those companies. Okay, because they didn't want to make $40,000 a year being a professor. Okay, bottom line is that I have an inside track. I go out to lunch with them all the time, and they have to look like this and say, "I never said this to you," but okay, the big boy chemicals that are worth over $100 billion just in the United States every year, the amount of resistance that they are incurring is through the roof. Chemicals that they have put hundreds of millions of dollars into have made $1 billion or more selling 2 years from now are worthless. Worthless. They're using chemistry, what is called vertical genetics, targeting a very narrow spectrum of the physiology of a fungus or an insect. And Mother Nature's a bitch, okay? She goes out there and she spreads her seeds around, okay? And 99% of them die, but 1% of them say, "Hey, that shit you just sprayed on me ain't doing nothing."
I like
And
it.
then it starts multiplying, okay? I'm not mentioning the company or the product. In 1982, I was the first one to publish a paper in Science illustrating scientifically, experimentally, that the most valuable fungicide in the United States, which had been on the market for 2 years, was now facing massive resistance in nature, and the natural public, that the company wouldn't talk to me anymore. They actually called Science and tried to block the publication. Okay, that's how nasty these people can be. It was published. Okay, I'm not proud of that. Okay, I'm not saying I'm there to destroy chemical companies. That's not my job. All I'm telling you, and we'll talk about this later on, is I have the inside track of knowledge. Every one of those major corporations I just referred to, all of them are putting hundreds of millions to billions into biologicals research. Biologicals to attack insects, to attack fungi, to attack nematodes. Okay? I predict in 5 years, you are going to see the most massive paradigm shift in the history of agriculture, where people are going to be using natural products in order to, insects, diseases, nematodes aren't going away.
No, they
It's
keep
part of nature, okay? It's part of nature that's been evolving for billions of years, okay? On the other hand, we have people to feed. Okay? We have incredible amounts of food that have to be produced and fiber, okay? But the use of chemicals, the age is going to be over due to, again, the idea of more and more data coming out with cancer clusters and problems that are directly related to chemical exposure, plus the idea that nobody is going to put $1 billion into registering a new chemical, knowing that 2 years from now, it's going to be completely resistant on the crops that they're targeted to.
So
So
we do a
it's
lot of,
exciting.
yeah, we do a lot of talk about horizontal gene transfer,
Yes,
biosymbiosis. These are the keys for nature because she can have generation after generation in a very short period of time.
very short periods.
So they can evolve way faster than these chemical companies can even create the molecule. Now they
Correct.
create the molecule very fast. And something
Correct.
interesting you said earlier was like, oh, well, every year they come out with new stuff. They come out with stuff every few months.
Correct.
And
Correct.
the scary part is what they have to do. They just change one molecule, and the old poison can be marketed as a
A new compound. Right.
safer new, even though it's a lie. But you know,
Uh-huh. I
it,
know.
the power of money and et cetera. They can just do whatever they want. But
Well, there's a lot of money there, my friend.
oh hell yeah, there is. And so and it goes back to like the Ford Pinto.
Yes.
The number crunchers realize, well, it's cheaper for us to get sued and pay out than
Right.
it is for us to replace the gas tank.
The,
So
the
let's
famous
just
cigarette, the
let it
famous
roll.
cigarette lighter. Yeah.
Yeah, let it roll. Let it roll. We'll take our hits.
Well,
But
again, I come from a school that puts out, I think, the second largest amount of chemical engineers every year, and a lot of them go to ag chem, and I know them, and I say, Jesus, Billy, what the hell do you do? What do you do? Okay. And he laughs and says, Jesus, Bob, I F around with molecules all day long, and I come up with new things in test tubes, and I send it to the plant people and the insect people, and they spray it on something, and they come back to me and say, holy crap, 1457-1112 was a real ass kicker on aphids. And they go, fantastic, we got a new product. Most of the time, okay, now AI is going to change a lot of that.
yeah,
But most of the time, they don't even know what they're doing. They're just throwing a new hydroxyl molecule onto something that exists. And 99% of the time, what does it do? Nothing. 1% of the time, it kills something ex post facto, new product.
yep.
So that age is rapidly coming to an end.
No,
And
and it has to. It
oh,
has
it has to. Correct.
right?
Correct.
So I mean, and the beauty of the direction both you and I are going
Mhm.
is that we've learned how to heal soil, right?
Mhm.
Not just turn it into something different, right? We're taking
Correct.
dirt and
Correct.
installing an entire web, a food web that will
Yes.
continue to expand and evolve, right? I don't know about you, but I get to see for the last 15 years anyway.
Yeah.
I've been able to just watch the ecology come back in. And it's
Yes.
the most beautiful thing in the world, right?
Yes.
First the plants come, then the insects come, then the birds come, then the pollinators come. Before you know it, you've got bigger and larger, more, big.
And it's been proven 100 times over. Correct.
Right. And it's just so rewarding to be there in that position for a job. And let's face it, this career is going to explode in
Oh,
the
yes.
next 5 years.
Yes.
If
Okay.
we
Let me tell you. Let
if
me
we
tell you
make it
something.
past 2030.
Let me tell you something interesting that falls right into what you're saying. One of my personality defects is that I study a minimum of 3 to 5 hours every night. Get to bed at 2 o'clock in the morning. Very fortunately, I maintain library rights at NC State and Duke. Okay, and I'm able to get. I have the passwords to go in because everything's online. Okay, what's a library? So I'm scanning the literature just constantly, and there was a paper cited in. I was published in PNAS, National Academy of Sciences, I think 2 months ago, that was referring exactly to what you're saying, called the consortia effect. And what they did was they did a long-term ecological study looking at the robustness of soil microbiota. Everything from earthworms and copepods to fungi to mycorrhiza to rhizobacters, okay? And they had all kinds of statistical ways of looking at it. But the more species that were present and the higher the titers of those species in the soil, the healthier the entire system was. more diversity of the biodiversity. Okay, the number of plants. Okay, I love talking about this because I have spent months there. There's a forest in the Peruvian Amazon that's way out in the back that was studied by a number of the most famous ecologists in the world. Okay, and the thing that always blows my mind, it blew my students' mind when I discussed it. And I have pictures of it. And I used to take them on field trips. We'd go down for 10 days and spend time there actually doing species surveys. There's this one area, a 1 hectare square, 2.47 acres. Okay. 2,197 species of higher plants on 1 hectare. Okay. Okay. That's beyond comprehension. Okay. Every moss, fern, to the philodendrons climbing up to the trees, to the shrubs. Okay. It is mind blowing. And what they're discovering now is that it is the consortia of soil organisms that promotes that kind of diversity. And you're going to hear more about that.
Yep.
But that comes back to agroecology.
Mm-hmm.
It's the same story. Okay, you go to the Midwest, we have been poisoning those soils since really it began in the 1930s with deep plowing and everything, but World War II, you know, God bless the Nazis. They discovered all kinds of compounds, which is the basis of half the chemical pesticides that exist today. It went vertical. Chemistry, better living through chemistry. Okay. God bless Norman Borlaug. I've met him on a number of occasions. He passed away a few years ago. The Green Revolution. What was the Green Revolution? Deeper plowing, more irrigation, more fertilizer, more pesticides.
Yep.
That's the Green Revolution. Okay. And the amazing thing that he accomplished is in third world countries, he quadrupled, quintupled yields by using his manifestations. And he didn't know any better. This is back in the early 60s, Ford Foundation. Bottom line is he ended up screwing up global agriculture.
Yes, he did.
Depending on it, and I don't blame him. You have to put everything in context of the time that he did it, okay? But no, those are not the solutions to optimizing yield and quality in world agriculture. It's a way of destroying the soil, okay? Most of the soil that I get samples from Midwest big farms, commercial farms, for lack of a better word, they're filled with nematodes, parasitic nematodes. They're filled with pathogens. And for the rest of the story, they're dead. There's nothing else there. They are literally dead.
Dude,
okay.
Same here.
I can isolate everything from the soil other than virus. Okay. And when you isolate and there's nothing there, okay, that means the soil is dead. And why? Because for the past 50 years, they've been putting 400 pounds of nitrogen down. They're putting their nematicide down. They're putting their pre-emergent, post-emergent herbicide down. It goes on and on and on. And I don't blame them. Why did they do it? Because they were told to do it. This was American agriculture. Okay, and all the companies would send their representatives out and say, we got the best damn new thing. You can't imagine it. I had somebody text me the pesticides they were using a couple of weeks ago. Okay, this happened to be on turf grass. Okay, it was 9 different pesticides. 7 of them I never heard of.
Yeah, they're all coming out with new stuff.
I never, what's that? And I go to, type it in and go to the MSDS sheet. I said, when the hell did blankety blank come out with that? Okay, but when their rep goes to and visits the people and say, this is the best damn thing, they have been enculturated to believe it. And you know, there's a place in you know where for people that are selling snake oil. And unfortunately, a lot of the chemistry that is out there right now really is snake oil. It's not benefiting anybody, particularly the farmer, who I have tremendous respect for. So
No, it's a brutal world as far as that's concerned.
I know
It's greenwashing, it's money, it's programming, it's
too much money.
lying,
Too much money.
it's cheating, it's stealing, it's all of the negative things that humans can do to each other.
Okay, so I'll tell you another quick story. And again, this is true. This is true because it sounds so ridiculous. It can't be true. Early on in my career at NC State, I visited, I told you that in the triangle here, we have all of these companies. And I visited a company who had a director of a person. I met him before when I was up in Ithaca at Cornell. I met him several times. He was down here, so I went. He took me out to lunch. Okay, and he says, "Yeah, I'd really appreciate if we can give you some money to work on this." And I said, "Yeah, okay, well, we could talk about that." And long story short, he had to go to another meeting, and I was around a board table with a bunch of their scientists. But one of the people there was their formulator. He was an engineer. He was the guy who took the chemical, mixed it with the adjuvants and the solvents and the stickers, and put it into a 1 gallon bottle. Okay. At that particular time, and this was a killer fungicide. Okay. The fungicide was going for $295 a gallon. That's in 1981 money. Let's be clear about that. Okay. $295 a gallon. I looked at him and said, "Look, I'm not telling anybody, but how much does it actually cost you? I'm not talking about the research and development and all that stuff, but to manufacture and produce a bottle of this stuff." You know what the answer was?
Pennies.
25 cents.
Yeah, pennies.
So when you start throwing numbers like that around, 25 cents to $295 retail, you know the rest of the story.
Mhm.
It's just too damn tempting. Okay, regardless, as you say, of the real facts behind it. By the way, 2 years later, not only was everything resistant and it was taken off the market, but the carrier that they were using was identified as being carcinogenic and they had to change it.
Yep. No, and the scary part is they can roll them out faster than they can be tested.
Correct.
And therefore, because they're not tested, we are their guinea pigs. So they get what we'll call special permits to say, hey, we've got this compound, it's doing all these beneficial things.
Exemptions. Yep.
Yep. And we'll come back with the data when we have it, in a year or two from now. Knowing darn well that 2 years from now, it's going to be stamped carcinogen or pathogen. So the game is real, and they're winning it. And unfortunately, there's not a lot we can do about that.
I know. I sat on a state committee that granted exemptions. And that was one of the ugliest experiences I ever had.
Yeah.
Okay, just knowing that all they're trying to do is sell material, but
and screw the population. To hell with health. To hell with nutrition.
And, I'm a capitalist. I'm not against companies making money.
No.
But the amount of fraud that goes into a lot of this stuff is really quite amazing.
All right, let's try to spin this into a more positive direction.
Yeah.
We could go down the negative rabbit hole the rest of the life, right? So let's wind it back into, okay, so in my experience in working with potatoes, we're going to go back to nematodes. I found that a very interesting situation where basically the farmers were being given misinformation about reusing their water, their cleaning water, reusing or not sterilizing plows between fields, and basically reinfecting themselves over and over and over again, and keeping their soils anaerobic instead of moving them to an aerobic environment, which the nematodes couldn't survive, right?
Correct.
And
Correct.
so when I got through the process and I sat down with the gentleman, I said, look, your SOPs are wrong. This is why you can't get rid of them. This is why you're using a different and more powerful nemicide every single year.
Every damn year. Yep.
And so you've got to stop the damage, because you're just never going to find the end of it through this way.
Yep.
And he's like, well, can we do a demonstration? I said, hell yeah, this is not hard. This is basically mimicking nature.
It's intuitive.
And moving away from your standard operating procedures, which is causing this downward spiral.
Yes.
And so he did a 4-acre plot.
Mm-hmm.
We proved that by not allowing the recycled water to go back on that field, and he was required to use fresh water. He was required to add a ton of organic matter into the soil, fluff it up and get it more aerobic. He was required to use new seed, new bud, not pulled from the old fields that was already infected.
Right.
And he had a bumper crop. Yep. Right. He knocked it out of the park.
Yep.
Very next year, right back
to the same thing. Yeah.
I don't understand. I've had this happen: golf courses, cornfields, beans, potatoes.
Absolutely.
And it's the most frustrating thing in the world that you demonstrate that here's a better way. How do you do it?
I don't have the answer to that, my friend, but what I suspect is what I said a few minutes ago.
Yeah.
You've got sales agents, you have county extension agents, okay? You have the man, okay? The man knows how to do agriculture, okay? And farmers are just like anybody else. They're hardworking SOBs. On the other hand, they're scared. They're scared, and they go running back to mama. Quick story, and then I'll tell you a better story. I'm going to ask you a pop quiz, and I'm going to ask for an answer for 5 minutes from now. Right. On a per acre basis, who uses more nematicide than any other, quote, crop? Okay, so think about that. So I'm at Cornell, and I am on the cutting edge of epidemiological science with potatoes. We're using, back then you had to program a computer with Fortran, with cards. That's what I was spending my nights doing. And we came up with models to predict incredibly accurately late blight epidemics. Okay, and I went to a meeting. I'm a kid, brand new doctor dude. Okay, and people were so impressed at Cornell. We have to invite you to this meeting to give this presentation. And I drive down to Steuben County, which is the large potato growing county. There were probably about 500 farmers there. It was an annual meeting type thing. And like a friggin' idiot, okay, I got up there and started talking science. And talked about, this is what our model said. If we do this, it gave us that answer. When we do that, it gave us this answer. But here's the bottom line, guys. You could save an incredible amount of time, labor, diesel fuel, and pesticide if you learn how to use this model, and we'll teach you how to do it. I almost got laughed off the stage. It was one of the most humiliating things I ever did in my life. So I go back and I sit down, and at the end, this guy comes up to me, puts his arm around me and says, Dr. Bruck, I want to apologize by some of the behavior of my friends here, but I want you to understand something. I said, yes, sir. He said, I'm a 5th generation potato farmer here in Steuben County. My great-grandma, grandfather, my father, and my sons will be growing potatoes. I want you to understand this. We know about late blight. We know about early blight. We know about nematodes. But religiously, that's the word he used, every Sunday morning before church, the family would get up at 4 o'clock in the morning, fill up the tractors, the sprayers, the spreaders, and we put Manzate and Temic and herbicide up and down the rows of our fields until it's time to harvest. Every Sunday, like the gospel. And I'm sorry, but we get a crop every year, and that supports this family. And I don't think we're about to change doing that. That was an important lesson. This was not a bad guy. Okay. He was a damn hardworking farmer trying to make a living. And through the years of the chemical companies and the county extension agents telling them, if you want to be safe, do this, and they do it. And that's a problem that exists a little bit less today, but it still exists. And we are going to be on a learning curve as these new biological products come out to get these people weaned off their addiction of having to go and put these things out there, to changing the paradigm to doing something new. Okay, as I told you, we are at a turning point right now. Is it going to be easy? No. But I really believe, because remember, when the big boy companies start putting out new biological products,
That
they're
changes.
going to put hundreds of millions into advertising it and spreading the word and everything. That's going to change the whole paradigm.
So let me answer your question. Potatoes, number one nematicide use.
No, it's
No?
very, but no. Okay, I'm going to give you a hint.
Okay.
Okay, it's a sort of an esoteric hint. As you said, I have a 1,000-acre potato field. I'm in Idaho, and it rolls. Okay, and there's high spots and there's low spots. And of course, nothing likes water more than a good parasitic nematode.
Yep.
They love water. Okay. So what do I do at the end of the season once all the potatoes are in and all the crap is out of the field? What do I do?
Well, you treat the low spots.
No, what do I do before that? It's getting the winter time. I got to do what do I do to my field?
Till it?
Yeah, with the same tractor going back and forth and up and down. I'm spreading the nematodes all over the field, from areas with a high concentration to areas where clods of soil sticking to the tines or to the discs are now moving it all over the field. Now, I'm going to give you the hint to the answer. And boy, have I done enough research on this to make me as frustrated as I could be. What do relatively well-heeled people do with spikes on their shoes?
Spread nematodes. So golf courses?
Yeah. Every golf course that I have ever looked at, and I've actually insulted certain people by saying this, is, geez, Steve, you got the best damn worm farm I've seen in all Florida. Basically, it's the same story, is that areas of a golf course will eventually get nematode problems on their turf. And those are serious problems. You got nematodes in there, you can wipe out a fairway or a green in a matter of days, okay? You got these guys up and down and left and right and this and that, okay? And they're pulling clods of dirt on their spikes. And given a certain age of a golf course, you have basically inoculated the entire damn golf course with nematodes. Then they discover it, and what do they have to do? They're spreading nematicides and spraying it once a week, every week, just to try to slam it down. But it's the same deal. You're using chemicals where you're never going to get 100% of them. And the ones that overcome it with resistance are simply going to grow back. And the guys are going to be marching up and down to this green and that green, simply spreading the nematodes. Remember, the average nematode is 1/4 of a millimeter in size. Okay. They're microscopic, and they can be traveling all kinds of distances on soil. So yeah, it's a major problem. And
Let's
the
not
answer, the answer is to make the soil,
help.
okay, to give the soil antibiosis against the nematodes so that they can never build up titers to where they can attack the grass to the point of killing it. That's the solution.
So let
From,
me share
yeah,
a story with you.
sure.
It's about getting laughed off the stage.
Yes.
So I put on a conference that actually was pretty much across the United States, and we ended up doing one in BC as well, called The Science of Organic and Regenerative Cannabis Cultivation. This was back in about 2018.
Okay.
Cannabis was legalized here in California,
Yep.
and Oregon and Washington.
I'm familiar.
So it was a big thing. Now I had transitioned out of very, very high end landscapes, commercial parks, national parks, all this crazy work that I was doing that just completely burned me out.
Right.
And I went on a cannabis cultivation podcast. Another person heard me and was like, "Damn, we need guys like you to come teach us how to do this." And I'm like, "Okay, all right. Well, let's put on a conference, because you can't teach if you can't see the people's eyes. Like,
Correct.
I could be going over your head, and you're just rolling your eyes because you don't, right? You know what I'm talking about. Teaching is more about paying attention to who you're teaching
Yes.
than it is about subject matter, because you have to sometimes slow it down or repeat yourself or
read your audience. Yep.
Totally, right?
Yep.
So we're doing this conference. I started in Portland, Oregon. We had a decent crew up there. There weren't as many legacy growers or OGs. These were a lot of young guys that were, money hungry and wanted to friggin' grow weed and make millions of dollars. So we weren't dealing with highly educated farmers.
Okay.
Group of 3 girls from Humboldt County came up to me after the conference and was like pulling on my arms and saying, "You got to bring this to Humboldt County." I'm like, "Humboldt County? Dude, that place is famous. You guys
You've been growing pot there for 100 years.
Yeah, when I was a kid, I would get the killer green coming out of there. Yeah, sure, whatever you want to do. When do you want to do this?" And like, "Well, can you come in a few weeks?" And so I went around to each one of the teachers that I assembled. I had Efren there, I had Dr. Elaine there, all the heavy hitters, right? And I asked them all, "Hey, can you guys, can you do this again in like 3 or 4 weeks?" And each and every one of them was like, "Well, yeah, I guess so, sure."
Sure.
So I went back to the girls. I said, "All right, you're on. What we need from you is a place to host us,
right?
Because I don't know the area, and so I'm not gonna have a lot of time to do research on where I should set up. Plus, Humble's pretty sparse, right? There's not a lot there.
Well, forest, yeah.
Yeah. So Wendy took the lead, and she came back and said, "We got you the octagon." I'm like, "What's the octagon?" She goes, "Oh, well, it's where we put our students, our kids. It's K through 12, because we don't have a huge population of kids. So it's like our school." And I'm like, "Oh, okay, that sounds cool." So I roll up, and I always led the path. Like, I started because I had soil engineering,
right,
and soil biology. So I was like, I'm the broad spectrum of the whole 3-day conference. You start with me, and then you build on each section of it. So I get up there and I start talking about soil horizons. So the O horizon, the A horizon,
basic soil science. Yeah.
Right. Basic, just understanding of how soil functions.
Right.
And I'm explaining, like, I've developed a system for you guys that will help you with your watering. It'll prevent the pots from going hydrophobic. It'll maintain the moisture level without you guys, freaking out and trying to keep underwatering and overwatering, blah, 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 blah. Laughed off the fucking stage. Yep. What do you know about cannabis growing?
Yeah,
and I'm like, and my answer always to them was, "Well, I don't care about cannabis. I do all plants."
Exactly.
Cannabis is just
No difference. No difference.
Cannabis is easy to grow. It's just a weed. Like,
Yep.
yeah, you want to push it. You want to get higher terpene profiles. You want to get more
Right.
THC or more CBD, whatever you want to get out of it.
Yep.
This will provide you the foundation to express the minor cannabinoids that you're not getting right now.
You got it.
You're overfeeding it, right? And so, like, most of them just got up and walked out. They're like, you don't know what the fuck you're talking about, kid.
Yep. Okay. I hear you. Let me tell you a story quick. Okay. And we'll talk about this more next week. But I have experimented with our products right now that we have. Our flagship product is called Rhizol. That's the one with 40 different microbes in it. We now have a spin-off product called Zenra for cannabis. Okay. Specifically formulated, very easy to use. It's a pill that you put in either the water or the soil, and bingo, it's done. What's fascinating, we have experimented with 43 different crops. The one crop that has responded by far the best in every metric you can think of, okay? Total biomass, root biomass, number of colas, weight of colas, extract of THC, CBD, CBG, terpenoids, cannabis. Cannabis had a greater percentage response to, as you just said, boosting the microbial feeding in that soil rather than fertilizing the hell out of it and spraying the hell out of it. And we are able to grow primo crops with this product.
Well, that was the other thing was I was explaining to these guys was like, you're obviously not hearing me because I'm giving you the perfect soil metrics to hold the biology.
Yes.
Like, you OGs knew about planting your cannabis underneath the trees. You didn't know why it worked so good.
Right.
That was the fungus.
Right.
That was the relationship to the forest and all the soil microbes.
Yep.
You dig a little hole, you throw some potting soil, you throw your plant in there, and all you had to do was water it. And sometimes you would load it with nutrients so you didn't have to go back and feed it again. But that was the reason why, was the microbes were breaking your nutrients down, making it plant available. Then when camp came down here and you guys all rolled into your friggin' underground containers, you couldn't grow anymore. And you would literally admit to me, I used to be a great grower. No, you were working with microbes and you didn't know it. Now you're working with chemistry and you can't grow like you do with microbes. And so there was that whole debate, and dude, I did it again. I got laughed off the stage again. But just as of recently, I think we've got our third or fourth major cannabis grow using the horizontal soil systems and using microbes and knocking it out of the park. Absolutely knocking it out of the park. So, you know, every good story has a happy ending. And
it's education. It's education.
It's all about education. And listen, you know what? We have 10 questions. So typically I break at an hour and a half to start answering questions. And the reason why is because the questions often lead to further conversations. So we know we got all next week. And next week, we're going to really dive deeper on a lot of these really very deep conversations. So Ken, why don't you hop on and let's see if we can get through these 10 questions in 45 minutes.
Oh, yeah. Okay. First one is from Farmer Cheryl. How much funding does Bob get?
Okay. When I was in academia, I got enough funding to stay alive, but it was not what you'd call hundreds of millions of dollars. Our company, ProGro, just started up 3 years ago, and we have done an amazing amount of work on pennies, keeping our lab funded, keeping the research going, keeping the trials going, paying farmers. Well, I'll just give you a hint. Back in 2023, we had 27 states running trials on over 70,000 acres, We're still trying to analyze the data, okay? I was responsible for doing all the pathology and nematology and all that stuff had to get done. But we are not that well funded. We are looking for funding right now. We think we got a tiger by the tail, okay? And as I said, this is the direction things are moving. So if Cheryl would like to buy some stock, she can get up with me anytime she wants.
There you go. That was a great way to end that.
There you go.
We're on the up, man. We're on the up. Just get on while it's low. Get on while it's getting's good.
Yep.
Well, her next question is: the number of viewers are citizen scientists. How can we fund our various soil and biology interests?
That's a really good word. You know, as a citizen scientist, you don't have a lab, which is necessary for being able to get all of this stuff that I'm talking about. You've got to isolate, you've got to identify, you have to purify. Next week, we'll talk about how we make our product, okay? That's not an easy thing to do. There's a lot of technology involved with it. What I would do is I would find a source of microbes that you trust, that you believe is going to do something good, whether you're working in cannabis or a home garden, it makes no difference. What we just said before, a green plant is a green plant, guys. Okay? You know, it's not like, oh, you have to have a very specific thing on corn. If you don't have this, then nothing's going to work. Doesn't work that way. But I would try finding a product that you trusted, that you can afford, and start using it. And the best way to convince yourself as a citizen scientist, let's assume you got a big garden. Cut it in half, Cheryl. Treat one half of it with the microbes. Don't treat the other half. Just use the regular, whatever fertilizer, whatever you're using, and look at it. Take a look at the quality, the amount of vegetables that you're getting out of it, and you'll answer the question yourself. So the science, once you get to that point, is pretty straightforward. Treated and untreated, see what happens. The science to get to the microbiology to make that happen is not something a regular citizen can do.
And I'd like to expand on that too.
Please.
You know, it's not only finding the microbes that you believe or trust that are going to help you. It's again going back to diversity. How often do you hear me say this? So maybe the real answer is to introduce those microbes to your compost, or introduce them and the compost, your indigenous microorganisms. Because again, at the end of the day, it's all about horizontal gene transfer, biosymbiosis. It's about letting the extreme phytes or the specific mycorrhizae to interact with your microbiome. And that's how you're going to get the great results. Go ahead.
Great answer. And remember what I said over an hour ago. Microbes feed on carbon. What's carbon? It's mulch. It's organic matter. It's, you know, everybody in my neighborhood looks at me like I'm crazy. I go and I pick up their leaves every fall and I throw them on my garden when they put them on the street to get picked up. And they say, why are you doing that? I say, that's fertilizer. And of course, they look at you like you're nuts. But that's exactly correct. Okay. A good mulching to any system is a fantastic idea, because not only is it going to feed the microbes, but it's going to increase the diversity of the organisms that are in there.
Yeah, leaves are supposed to be left. Leave your leaves.
Yeah, remember something that's we haven't talked about at all, but they're really, really good for soil. They're called earthworms. What do you think earthworms eat? Leaves, weeds, organic matter. Okay, so we remove it. We got, that's trash. We got to take that away, and then we spend $500 on fertilizer, putting it on our lawn. Whatever.
Bob, you know what killed me when I moved to California? The green waste bins.
Oh yeah.
Everybody harvests all the green material off of their property
and stack it up
to get rid of it, and wonders why their soil's so depleted. It's just beyond me, but I won't go there.
It's free fertilizer. Bring your truck up, baby.
All right, let's stay focused here.
Let's focus.
Ken.
Okay. Well, I wanted to, and I can't remember the name of the damn thing. You put the paste on, you cut up your leaf, and you look through it under the sun, and you
it's called the Brix test.
Yeah,
Brix
meters.
Sugar.
Yeah.
Brix goes up in grapes that are inoculated with rhizol. Yes.
There you go.
Okay. It goes up in hops. Okay. We actually wanted to get a trial with sugar cane. We couldn't pull it off. But yeah, actually microbial working with the roots and everything actually increases Brix rating.
So this gentleman that I've had on the show a number of times, Michael Whitman, he's the one that sells these brick meters.
Yeah.
He makes a biochar mixed with worm castings and a few other microbial components. And the highest Brix levels he's ever achieved was 17 on, I forget what the plant was.
Yeah.
We've hit 22 with one or two of product. Yep. So, again, and I try to talk to him about diversity, and he's so like got the horse blinders on that he doesn't understand. It's like, dude, you can get higher than 17. You can get into the upper 20s.
Yep.
If it's all about diversity. So anyway, that was a side tangent.
No, that's very well done. But there are great experiments talking about high organic matter, high microbial populations, actually increasing Brix readings where you want them to be increased.
Yep.
And that's money in the bank for things like wine. So
for everything, it's no pesticides. It's better vitamins and minerals and nutrition. It's everything. So anyway.
Well, I just wanted to point out that by using a Brix meter and having it slowly go up, you're actually being able to track how much activity and diversity is in your soils. So Cheryl, there's a good, very cheap method to track, as a citizen scientist.
Yeah, it's
$20 on Amazon. Get a Brix meter. Yep.
Actually, there's a better one. Go on to Blue Sky Biochar. It's $133, but it comes with a scissors. It comes with an actual grinder that you can get sap out of. The cheap ones, you're just going to be working and working and working.
Right.
Trying to get something out of it. You're going to get frustrated if you don't break the damn thing. So
that's an experiment that I actually had somebody do. Is every week they would go and pull samples and take a Brix reading and see what was happening in treated versus non-treated.
Mm-hmm.
That's good citizen science right there.
There you go. It's all about the data.
Yep.
And that's why I wanted to point out the Brix meter, and it's Blue Sky Biochar. And like the scissors he sells, it's a 5-blade scissor. And he's got the press. He's got sheets on different ratings for Brix, etc. So it's a good place to start as a citizen scientist.
And I took his press and I beat the shit out of it. I was squeezing compost, like not fresh leaf material. I was trying to get liquid out of compost. I got it. I did get it, and I didn't break the damn thing. So I'll stand behind that product.
There you go.
Yep. Okay, our next question from Cranberry Cass. What do you know about Iroquois? Yeah, I can't even pronounce it. Leighton, can you pronounce that?
Iroquois mycorrhizae. We didn't talk about Iroquois. Iroquois is, I think it's rhododendron, azalea. It's a very, very small fraction of plants that actually work with Iroquois.
Yes.
I don't know, Doc, can you talk a little on that? Do you know
what you're talking about, plants from the heath family, that have endomycorrhiza that are very specific to things like rhododendron. Blueberry is in the same family. It's heath family, Heath. I have not personally done a lot of work with them, what we did do were trials with Rhizol on blueberry. Blueberry's a big crop here in North Carolina, and we got some fantastic results in terms of yield increase, Brix readings, okay, and suppression of several nasty soil pathogens like verticillium and fusarium that attack those soils. So I've done some work, but not a lot.
Would cranberries be in that family?
Yes.
Yeah.
Okay.
Correct.
So the interesting thing too is that in the right fungal dominant soil, fusarium actually becomes somewhat of a beneficial. It's not.
I've seen that.
So,
Correct.
Again, Cass, the best way to get your Iroquois to go to that bog that you have that was in nature untouched for years.
Yep.
That special one that's way up in I forget what park that's called, the Miles Standish Park. That he knows what I'm talking about. Hopefully he's still on here. Go up there, and what we would encourage you to do is look up natural farming. The foundation of natural farming, or Korean natural farming, is IMO collection boxes. Now, you're not going to capture the exact mycorrhizae that you're looking for, but what you will capture are the prerequisites that fungi needs to be present in order for it to propagate, in order for it to sporulate, right? So then once you've got that IMO collection, you can start to mix in some root fragments from the berries that are existing up there that have not been poisoned.
Right.
And you mix the root fragments in there, and you let that work together, whether
they'll grow.
Or expansion, like when you do a IMO 3, right? But remember, you got to do your sugar stabilization first, so you don't lose your IMO. Once that IMO 2 is done and stable, now you can start playing it with the root fragments, and then start growing it out with your IMO 3. And now you'll have inoculant, more than enough inoculant to do whatever bog you want.
Correct. Good question.
Bob, would your product be beneficial for the cranberries in that nematode direction?
Yes. Yes.
Well,
as I said, I have done work on rhododendron, azalea. These of course are horticultural crops where we live. Okay, people plant them all the time. And as I said, we did an actual experiment with the university with blueberry, and we saw some very positive results.
Yeah, Cass, I would say definitely, reach out to his website and basically explain what you're looking for. And he's got the people that will respond back. He's got a number of different products. You may want the shotgun, or you may want the sniper product. I don't know. But that's something I'd reach out to his crew and his consultants to tell you.
And we'll talk next week about specifics about what these things actually do. Yeah.
That means you got to come back next week, Cass.
And Cass is watching from X, by the way.
Oh,
X. Okay.
Yep.
Say hi to Elon for me.
The next question, Doc, is when you look at the ground, do you see plants or do you see fungi?
That's a great question, okay? I'm afraid that I am jaded, okay? Every time I look at a soil, I immediately click, wonder what the hell's in there. So this is where people at TSA, and I go on a trip somewhere, and I'm bringing back little baggies of dirt, okay, which is so long as it's within the United States, that's totally legal, okay. But what's this? It's dirt. All right. So I'm an old man now, and I have a hard time. Okay, my daughter is the gardener. Okay, and she's looking at the plants and makes them grow like crazy. I'm pretty much looking in the dirt.
So you'll appreciate this. So early on in my work, I was still at Rodale. I was also working parallel with Dr. Fitzsimmons down at U of A.
Yes.
He has the aquaculture and what's it called, the environmental controlled research facility down there. Mm-hmm. And I was focused with him on basically fish maul, which is fish manure, but fish don't make manure. They poof. It's just organic matter and biology floating in the water column.
Poop out their food, yeah.
Yeah, mixed with their food and whatever, right? So, needless to say, I was down there and there was a monsoon the day before, and I've been down and back because my daughter was at U of A or ASU. So I was in a world of hurt because those colleges hate each other, right? I was getting shit on both sides as I was going back and forth.
I'm aware.
So anyway, so there was this, freaking monsoon, and I drove through the desert. There was desert, right? Then there was a monsoon. I drove back the next day. My daughter had some kind of event that she wanted me to attend. So I drove back up for the event, and I went through the desert. I'm like, oh my God, it's full of life, right? so I blown the fuck away, right? So
just like that. Exactly.
Just like that.
Yeah.
So I go back, see my daughter. The whole time I'm thinking, like, what the hell did I just witness?
Yeah.
And then I had to go back down to U of A the next day. So I got down and I talked to Fitzy. I'm like, Fitz, all right, I thought desert soil was the most infertile soil on the planet.
Mm-hmm.
And he goes, no, actually, Leighton, when they built Biosphere 2, which I know you love and talk about,
right.
You've been there a couple of times,
right?
They chose all desert soil. They could have got soil from anywhere in the world.
Lots of nutrients. Yep.
Yeah, they knew that the efficiency and the value of that desert soil is worth way more. Plus, it was right there. It was cheap. We didn't have to import anything.
Real cheap.
So anyway, so I'm like, all right, I'm going to collect some and bring it home with me. So what Doc told me was, all right, look, Leighton, be really careful, be really mindful, be respectful. There's this thing called the crust. And he goes, you'll see it when you walk out in the desert because when you step, you'll break through the crust.
Salt.
It's only millimeters thick.
Right.
But that's the key because that crust protects the microorganisms underneath it from cooking and drying out. So he goes, break the crust and harvest the soil right underneath it. If you see a footprint, don't collect it there because that's probably dead.
Right.
And he goes, I don't want you marching all over the desert trying to find this stuff because you're disrupting the whole ecosystem.
Correct.
So just get as close to the road as you can, step, and if you see that crust crunch, then collect it. So I collected 2 full baggies of it, threw it in my luggage, threw it in my backpack, and hopped on a plane. Well, I didn't get on the plane very easy. They were like, all right, kid. What are you smuggling here? Come on.
What is this?
Yeah, is this heroin? It doesn't look like cocaine. It's got to be, meth or some kind of
sand
or whatever. Right? I'm like, no, it's just dirt. It's dirt. Why would you be bringing dirt back home? Come on. They didn't believe me. Like, I threatened with a friggin' cavity search, dude. Like, I'm like, no, I'm gonna miss my brain. It's soil, man. It's soil. Taste it, touch it. So finally, they let me through. But man, was that
that's why I always carried my APHIS certificates with me wherever I went.
Well, when you said that earlier today, I was like, I'm such an idiot. I was so naive. So naive.
Plus, I mean, no one's going to believe you. Okay, I mean, it's just what do you mean you got a bag of dirt from the Amazon? Okay, but I actually outside of Phoenix, about 30 miles. Is where I collected a bunch of desert soil samples that I isolated from, and 2 of the organisms in our mixture are from that sample.
Love it. All right, so quick story on the Amazon.
Yes.
I went down and lived with a tribe of Indians for 2 or 3 weeks in Venezuela on the Amazon side or the Venezuelan side of the Amazon.
Right. Down the Orinoco or what?
Near that, yes.
Okay, all right.
So I got nailed by these little bugs. And I got back to the village, and I got swarmed by the women and children, and they were popping these little things all over my body.
Okay.
They were called pulis.
I've heard that.
And they're just a tiny little red dot. I mean, just like the head of a pin. Very, very small.
Yep.
Hurts like hell when you pop them. But they said, well, through the interpreter, because I had to speak to John I spoke English to Johnny. Johnny spoke
Spanish.
to the Indian we got out of the mission, and the
Right.
Indian spoke Spanish, or translated to Indian,
Right.
to the chief, and it had to come all the way back. And the chief basically came back and said, "Well, if we didn't do what we did to you, your skin would have basically been peeling off within a couple of days."
Toxins.
Right?
Yes. Yep.
And I had this conversation with Kevin Jodry, who was growing large-scale cannabis in Colombia, and he ran into the same thing, except for nobody told him this was going to happen. And he said his arms were oozing for months. So did you run into anything like that way up in the...
I did not run into pulleys, but I lived for 2 weeks with a Yagua tribe near the border of Brazil, Colombia, and Peru. And all I could say, and I think about this all the time, I have never met such healthy people in my entire life. That means, I'm getting chewed up by mosquitoes and bot flies and everything. Their skin is smooth as a baby's bottom. They all are well fed. Their teeth are intact. Okay. They're smiling and laughing all the time. When they say that these people know something that we don't know, it's true. It's true. There's absolutely no question about that. And the days of Schultes at Harvard going down and living with these people and learning about them are pretty much over. Now we're trying to mine for diamonds and gold and just, it makes me, I follow the Amazon very closely. And every time I see the new data that's coming out, I get nauseous.
Oh, I agree. It's almost tear jerking what's going on now.
The forest is going, the people are being assimilated. Okay. I was in one village and everything where I noticed something bizarre. Okay. And the students that were with me noticed the same thing. There were old men and women, and there were babies, old men and little toddlers running around. And I asked, where are the parents? Where are the teenagers, the young men and women? And they were just point blank said to me, all the young boys up to, 20, 30 years old have run off to the mines, and all of the girls have run off to the brothels that are in the mines. And that's how they make a living. I mean,
dude,
fuck that shit. Okay.
Dude, I saw
it's
the same thing.
just disgusting.
I saw the gold miners just polluting the river. I saw horrific things, and that was the
mercury, arsenic. It's just disgusting. Really.
That was the reason I went down there, was because I had been infected by a movie called The Emerald Forest when I was a kid.
I saw it.
And that drove me to want to go down there and see it before it was gone.
Yes.
I had no idea the devastation. I mean, we drove through hundreds of miles of deforestation to get where we were going.
Yes.
It was horrifying. It was absolutely horrifying.
It's called progress. Don't you know that? It's unbelievable.
Anyways, that was
I know that's another story.
Because, man, I got ripped up. I got ripped up by leaf cutters. I got ripped up by the polies. I mean, and we even went monkey hunting. So they only eat monkey once, maybe a year, if that. Yeah. But it's a celebration, right?
Yes.
And they taught me to shoot the poison dart, right? So I practiced. And the only reason they did this was because the chief's daughter had a cut in her foot that was super infected,
right?
Like really bad. So all I did was I'm like, all right, you guys hold her down. She's not going to like this.
Yeah.
Took out my knife, sharpened it, hit it with alcohol, and just basically scraped out all the pus and all the dead flesh.
Right.
And then I packed it with a little bit of gauze. I put some Neosporin on it.
Neosporin. She got healed.
Wrapped it and bandaged it. And I'm like, all right, when she goes to the river, when she goes to the river, you got to keep her foot out of the water, because the water's going to make the infection come back. Because they go to the bathroom in the river. That's how they go to the bathroom. Pee or poop doesn't matter. You go to the river, you do your business, you come out. So I'm like, you got to keep this thing dry. So within 3 days, it had turned pink it was pretty much clean. And fourth or fifth day, it was like almost completely healed. It didn't really even scab. And that was the reason why they celebrated. They wanted to celebrate me. They gave me a monkey tooth necklace, which I still have.
Sure.
I was a member of the tribe.
Sure.
So we're going out hunting, and they gave me the.
That's very prestigious. Very prestigious.
Yeah. You got to do the gun. And
yes.
So I'm like, I thought I was badass because I practiced for a few days, right? And I was good. You get out there and there's a monkey moving around, and it's called buck fever. You just get so excited. No. It's funny.
One guy
grabs the fucking gun.
Done.
He gives it to his fucking, his right hand man.
I've seen it.
Bunky comes down. Well, we're out there, and there was this freaking lion up on the hill on the knoll, and she's like, "All right, we got to get out of here right fucking now." He goes, "That thing's gonna come down and kill us because that was really, really mad. They've deforested this area, and he's not leaving, and he's pissed off, and he knows we're down here now, so we got to get out of here." So that's how mad the forest is at humans.
Well, I'm reminding myself now to send you a picture. Okay, about 20 years ago, I was with a group of 30 graduate students way upriver on the Napo. Okay, very remote. And one of our guides comes yelling up to our camp. It was almost sundown. He says, "Come, come, come." He's yelling and screaming. What the hell's going on? I have a picture that's been certified as the longest green anaconda ever photographed in Peru.
Wow.
24 feet 6 inches long.
Damn.
And we were right up next to it, maybe. And I'm saying, you people are crazy. Get the hell away.
You talk about food. We fit perfectly in that
belly.
Oh,
I mean, the thing had to weigh 700-800 pounds.
Anyway, probably a couple thousand.
Girth like this.
Yeah.
But I got that picture. I look at it at least once a year.
Now, did you run into any shit with the shining star in Ayacucho?
Oh, but you've got all kinds of stories.
Oh, I've been.
The last trip I took. Okay, our outfitter calls me up 2 days before we're leaving for Peru. And he says, "Look, legally and ethically, I've got to tell you this. Okay, but I don't want you to worry. I'm going to tell you what the solution is. The Sendero Luminoso is deep in the Amazon again, and they are targeting tourists for money, for passports, specifically passports. They love American passports and everything. I want you to come. I don't want you to worry."
Oh, no.
He's getting a phone call is what's going on.
Oh, okay. I thought he lost the power.
The bottom line is I am going to have a group of 4 armed guards with you 24 hours a day, whether you're at the camp, whether you're on the river, whatever. And boy, they were the real deals. Uniformed AK-47s. We never ran into anything. But that was 2 days before we were about to leave. We were told that there have been people that have been accosted deep in the.
When we got into Ayacucho, we were in the back of the bus, and we'd made friends with a lot of the college kids on that bus.
Sure.
And they're like, "All right, when we get into this village, you guys duck down, stay undercover, do not get off the bus under any circumstances."
Interesting.
And as we rolled in, we rolled past a number of headless, limbless bodies.
Are you serious? Oh,
yep, propped up on the side of the road. And these were Americans. These were gringos, because you could see they were white.
Yeah.
They weren't brown. So they're like, "Yeah, this is really, really dangerous place. We'll get you food. Just stay on the bus. You got to get through here. Once we get over the bridge on the other side, you'll be safe." And when we got to that bridge, we got ripped off the bus by the PIP. And they put Uzis to our heads.
Yeah.
on the edge of the friggin' precipice. We thought we were dead. We were like, oh, we're dead. This is
It's over.
it.
It's over.
And some little old lady got off the bus and started screaming and cursing at these PIPs and was like, these guys didn't do anything, blah, blah, blah. You got to let them go. You got to let them go. And, you know, a couple of the kids, the college kids, stuck their head out the window and they're like, they're not gringos. They're not gringos. You know, just like trying to support us. And finally,
Right.
was like, gave us our passports back and said, get on the fucking bus and don't you ever come back here again.
We got some war stories we can tell.
Oh, dude, it was.
The last one I'll tell you is we're in a very remote camp, and there were a bunch of dugout canoes, and the outfitters said, please, if you take out the canoes, just stay right in the area, because last year we had a very bad event. And we said, what's that? There were two Swedish tourists, blonde, blue-eyed, tall, white as hell. They decided they were going to grab a dugout and go way upriver, and they never came back. And the El Polizia went up to investigate and found their heads on a stake, okay, at an Indian village, in which they interviewed the Indians and they did not prosecute because the tales of this tribe was evil white men, devils, would come to steal your children. They came up in this boat and they blowgunned them to death. Cool.
Oh, we're going to have more conversations.
We're going to have fun about that. But needless to say, my students did not take the boats upriver.
Yeah. Anyway, we got to punch through 8 questions in 15 minutes. So let's
what we got?
The answers short. And we'll talk more next week.
Yeah.
This isn't a question, but I had to bring this up.
No sound.
Caught with soil in condom in
no sound.
Examination. I can see the headlines.
Oh. I didn't put it up my butt. I just put it in my backpack.
Ken, I did not hear any sound.
Okay, can you hear me now?
I still can't hear him.
Okay, it maybe you might have to jump off and back on. It's your phone.
Can you hear me?
I can hear you. I can't hear Ken. All right, tell me what the question was. Good.
It was just a comment. You can read it right there. It basically says he was making fun of me.
Okay.
Smuggling soil in my butt with a rubber.
There you go.
All right, so you know what? Ken, you ask the question and I'll ask it to him.
Okay. What's the best way to either feed or activate these fungi?
All right. Can you see that? It says, what's the best way to either feed or activate these fungi? And I'm assuming it's the fungi within your product. So I think he's trying to drive at. Do you have a food?
All right, just real quick. I can answer that question quickly. Okay, we grow our biomass using bioreactors, liquid. Then our engineer has modified a large lyophilizer or freeze dryer, very modified, patented, okay, to be able to take that liquid and turn it into a powder. And from that powder, we either have a bag of powder or we make pills out of it. And it is completely water soluble. So you throw the pill or you put a gram of powder in water, and within 30 seconds, it is ready to be activated, and it brings the fungi and the bacteria back to life.
And Leighton, just to let you know, he's on a phone, so he's not able to really see.
Oh, got you.
Yeah.
So the next, you can hear him though, right?
Yeah, I can hear him fine.
Okay.
Just you guys are.
I can only hear you. Yeah.
Yeah.
All right. So how do you recommend transplanting Iroquois mycorrhizal? Soil plug in a tea or something? That's Cass. So Cass, I would say that once you have grown out your IMO 3 using that collection and then using the root fragments, I would say probably the best way is to liquefy it. So a limo is what they call it, the liquid IMO. So just make a tea out of it. Don't add any foods because you might get a bacterial plume. You don't want that. You just want the fungi alone. So I
Yep.
hope that answers your question.
I agree with that. Just throw it in the soil. Yep.
Okay, Leighton, you can read this one.
Who's in charge, the plant or the fungi?
That's actually a nice question. And the answer is both. Okay. One can't do without the other. And we learned that lesson the hard way. Okay. You put a plant, I don't care what it is, in a dead soil, and you're going to have one crappy plant. Put it in a vibrant, living, biologically active soil, and they're going to work together to grow what we're interested in growing.
Okay.
All right. Let me ask this one. Hang on, Ken.
Okay.
I love to ask this question.
Hmm?
Where does the plant end and the soil begin?
Where does the plant end and the soil begin? I'll go with the soil became before the plant. Okay. Is that if you don't have the soil, the soil's mother's milk, for Christ's sake. Okay. That's the basis of a plant. It's getting carbon dioxide from the air and everything else from the soil. And you simply have a green plant with carbon dioxide alone, you don't have a green plant. So you've got to have that symbiotic relationship. And we can prove time and time again that it works.
So that wasn't exactly the way I asked the question, but that was one of the most intelligent answers I've heard. Typically, I say, where does the plant end and the soil begin?
Oh,
so
I see.
You've already got both.
Okay. Well, I was told once that when I was learning how to become a botanist, is when I do planting, is that the brown side goes down and the green side goes up. I said, okay, I got to remember that.
Well, it's kind of a redundant question because it doesn't. The soil and the plant are unison because of the fungi, because of the rhizosphere. There really is no beginning or end. They're
let's talk about something really quick. Okay, we keep talking about the soil and the microbes. One of the best way to screw up any type of a crop, from a garden to 1,000 acres of soybeans, is soil compaction. Okay, the friability of the soil, the ability of it to aerate, to percolate water, to have oxygen. Roots have to have oxygen. How many people know that? Okay, nobody. Okay, roots have to breathe. Okay, microbes aerate the soil. Their activity, enzymatic activity of breaking down nitrification, denitrification, mineralization, demineralization, makes a soil friable. So forget about all the biological stuff. Now you're talking about soil physics. The actual physics of the soil is greatly enhanced when it is enriched with microbiology. Period.
When it has, the microbes make the aggregates. The aggregates provide the functionality of the soil.
Correct.
Yeah, spot on. All right, go ahead, Ken. What's the next one? All right, how can biology help the microplastics problem? Can or should fungi replace plastics?
Okay, two answers. An Indian research group has just made plastic out of fungi that is completely biodegradable in 2 weeks. Literally, they hold up this plastic bag, like you go to the store and get the plastic bag. Okay, it's a little opaque. Okay, but you can carry 20 pounds of groceries in it. You throw it in your garden, and 2 weeks it dissolves. That's number one. Number two, MIT announced, okay, just early announcement, they published it in an abstract form. They now have a bunch of extremophile bacteria and fungi that completely digest microplastics in the environment. Hallelujah.
Yep. So the answer is it's coming.
It's
And
coming. It's coming.
very quickly.
Good question.
Okay. Next one, Leighton.
Would applying spent oyster mushroom blocks be a fix to eliminate the nematodes?
I don't think so.
I agree. No,
I don't think so. Yeah, oysters are of a group of Basidiomycetes that there are very few organisms in it that really have antibiosis activity.
So the real answer is change the soil. Get
Correct.
it aerobic. Get a lot of competitive microorganisms.
Correct.
Get some beneficial nematodes, predators. Or
I love your mulch pile thing. I mean, it's something that I've always wanted to do.
Oh,
And
dude.
I'm too damn lazy to do it. Okay.
Dude, you have no idea.
But all the crap that's in there percolating and cooking and everything is filet mignon to plants. It's
Dude,
incredible.
I have a whole compost kitchen. No, seriously, like I work out of this facility called Peach Hill Soils.
Uh-huh.
I have access to green materials, brown materials. Every day there's more and more stuff coming in. And I just go down there with my scoop or my skid steer and I grab whatever I want. I've got all these recipe piles, all these different piles for different products or functions.
I'm a bad boy.
Yeah, you got to come out and visit.
I will. I promise. But yeah, I mean, I've seen people. I used to have sort of a secondary mother who, after my parents passed, they took care of me. And she would have a pile of, giant thing in her backyard. She'd throw everything in it. Okay. And then she'd have a garden. And I said, what the hell are you doing in this garden? All I do is throw that crap in the mulch pile on it. I got steak bones and fish heads and vegetables and everything. It just cooks in there, and I throw it on. It grows like crazy.
And then I took that and put it back on the garden.
Right, man. And when you get down to it, it's not rocket science. Okay, it's really not.
So you really kind of answered this question on the tail end of the last question, but you might want to ask it again.
So, what is the nematode's worst enemy?
The nematode's worst enemy. Wow. I'll tell you. Right now, I'm going to just repeat myself a little bit. In areas that are heavily infested with plant parasitic nematodes, as of right now, is there anything I know of that will eradicate that problem? Going from the most carcinogenic chemicals down to biologicals, down to soil treatments and everything. I don't know of anything. Okay. If the problem is entrenched, you've got a real problem. But our preliminary data shows that a consortia of very specific microorganisms can, and here's the key word I use, bring the population down to be subclinical. Subclinical, meaning I can still isolate nematodes from that soil, but there aren't enough of them to cause enough damage, whether it's a golf course or a soybean farm, that I'm going to lose a significant amount of my yield. That I'm convinced of, that we are on the threshold of being able to do that. But back to the old thing, 99% of what's done is chemistry, and the chemistry is feckless. Absolutely feckless. I've seen places that they're applying this stuff every damn week. People, I mean, on golf courses, you've heard of these stories where people faint from the fumes being given off by pesticides. And the most of those pesticides are herbicides and nematicides. Okay, that's the greatest amount that's applied. And I have never seen a, quote, clean golf course, ever.
I actually have seen one.
Really?
In San Francisco, run on Korean natural farming. No herbicides, no pesticides, no nematicides.
Okay, and they have no problems?
No problems. Well, they have some broadleaf plants that are mixed in, right?
Yeah.
So it's not a manicured PGA course.
Right.
So
Right.
they've convinced the public that, hey, we can't run this golf course because it's right on this bird sanctuary watershed
Right.
any other way than this way. So if you want to play, you got to see a
Deal,
few.
deal with it. Deal
You
with
can
it.
see
Right.
a couple dandelions here and there, right?
Right.
It is what it is. But the key to the whole thing is, again, I would answer it as conditions.
Yep.
He uses a deep tine a lot. And he doesn't just put sand. He puts sand with biologically complete compost into those deep tine holes. So he's getting the aeration down in there. He's also using a ton of microbial biological inoculants that he makes on site with compost and IMOs. So he's eliminated these kinds of pests through environmental changes, aeration, and then biological treatments. So creating that diversity. And he's operating 100% clean. That's the only place I've ever seen that did it.
Well, that's a model, and good for them.
Okay.
Rumor has it, I have not followed up on it, but about a year ago, I learned we have beautiful mountains here in North Carolina. Okay. And a lot of very high class golf courses, private clubs up there. The word that I got on the street was that Tiger Woods was going to build a 100% certified organic golf course in the North Carolina mountains. No chemical fertilizers, no pesticides, period. So if he could pull it off, God bless him, tell you.
Yeah, I was in heavy negotiations with one of the heavy hitters on the PGA, the council, on just this subject. And I assured him that, yeah, we can do it. There's definitely ways to do it.
And he told you, go fuck yourself.
No, he was wide open. It's the rest of the board that shot him. Just said, no, you're not going to continue these conversations. That's the end of it. We are synthetic. We are going with True Green and all of our other buddies in the chemistry.
I have dealt with superintendents left and right. And you talk about reticence. Okay. These are highly paid, highly nervous people.
Yep.
Okay. God forbid at Augusta National, there's one blade of grass that's brown on the fifth fairway, someone's going to get fired. Okay.
I know.
So this is part of the problem with that whole thing. But so talk about a fruit ripe to be picked. Golf courses. You know how many golf courses there are in the United States,
Dude, I was very blessed. I got to work. I did work with some golf courses. I did turn them.
Okay.
But what happened was the new president came in, fired the old superintendent, and he went back to chemistry.
Right.
But anyway, I did get a chance to work.
Good for you. I've had
years with the Dallas Cowboys superintendent.
Right.
And we were 100% biological. He wasn't having fairy rings. He wasn't having any of those fungus. And we had beneficial nematodes. We did have a couple of root feeders, but we were able to keep them at bay because we had such high levels of predatory nematodes.
Yeah, well, back to that word again, subclinical. It doesn't matter if they're there if they're not doing anything.
Right, exactly right. So, and then unfortunately, guess what? The management shifted.
You got fired, right?
Brought in a new consultant, and we documented the destruction of the soil food web over 6 months. And I kept saying to Chris, I'm like, keep testing. Just keep testing. Keep testing. Let's document how quickly it could be destroyed because we know how long it took to build it up.
Sure.
And I want to be able, he's supposedly going to come on this podcast now that he's no longer there and talk about just that. But he's trying to transition into a new job.
Right.
And so he's kind of tied up. But eventually he'll come on here and talk about 4 or 5 years that we worked together.
Great.
Anyway, is there one more question, Ken?
Yep.
What is the best way to use aloe on cannabis? Is it to spray Epsom salt as a foliar? If so, how often? Well, that's 2 completely different questions.
Those are 2 different questions, and I'll tell you, spraying stuff on cannabis and not knowing exactly what you're doing can be very dangerous.
Hell yeah.
Okay. You can turn a beautiful flower, nice leaf and bud into a spotted mess. Okay. Because you have a combination of the amount of light that's going on it, interacting with chemical substances. Okay. So, I don't like to talk about that because I don't have enough experience, but you've got to be very, very careful because you could destroy a good looking crop in a very short amount of time.
So the way I would answer that is aloe is a surfactant.
Yes.
What it does is it helps water spread
Correct.
over hydrophobic materials.
Correct.
Your plant is not hydrophobic. If your plant's hydrophobic, that means it's dead.
That's correct.
There's no need ever to foliar aloe on a plant. Now, if your soil's hydrophobic and you want it to take the water up, that's where I would use aloe.
Yeah.
As far as Epsom salt, absolutely
No.
not. That's going to cause phytotoxicity.
Absolutely.
You're going to kill your plant because the sunlight's going to go through it.
Correct.
Epsom salt should only be used as a chemical or a chemistry added to your soil. But again, we're not into chemistry. We're into biology and organic matter and organic carbon.
You're going to change the pH of that by.
Yeah. I mean, look, the only limitation of biology is chemistry. If there's no molybdenum in the soil, I don't care if you got solubilizers that are present. There's no molybdenum to solubilize.
Yes.
So chemistry, yes. At the beginning, you always do your chemistry. If you have some deficiencies, get it balanced then.
Correct.
And once the chemistry is in sync and in the right place, that you have everything that you need, then add the biology, plant the plant, and sit back and enjoy the show.
Yep.
So that's the way I'm going to answer that. And
I've seen more cannabis in the field destroyed by over-fertilization.
Oh, God.
Because someone says, oh, geez, it's turning a little yellow. I'm going to dump 400 pounds of NPK on it. And that's it. It's over.
So we have a saying here. It's don't be a moron. A little good, that doesn't mean more is better, right?
Right. A moron. I got it. I got it.
Dude, I use that at the cannabis conferences. I got all kinds of dirty looks.
Oh, I'm using that one. I'm stealing that. Moron. Got it.
They're all bro scientists. They're all, oh, yeah, I know what I'm doing. I just add more. I'm like, no, that's going to exasperate the problem, you fucking idiot. And I'm sorry. I'm getting.
Well, not only that, but cannabis in particular. Once you're flowering and it's starting to set the cannabinoids and the terpenes, nutrition, over-nutrition is the best way to screw it up.
Yep.
You put too much nitrogen on there, you're going to have to smoke a pound of the stuff to get a buzz.
Well, worse yet, you're going to create newt lock, and it's going to taste like shit. It's going to get all cakey and nasty and resiny.
Yeah,
anyway,
so moron is not the way to go.
Exactly.
Yep.
All right, we're about
done.
8 minutes over, and I've got 2 puppy dogs staring at me like it's time for my walk, Dad. So I really want to thank you, Bob. It has been just
fun.
Pleasure. This 2 hours has flown by. We have so much more to discuss. We're going to deep dive again next
week. Same time, same place. We'll do it again.
And then we'll probably after that want to have you back again because I know we're not going to get where we need to go. There's too much for us to talk about.
Excellent.
So I want to thank
you guys. Thanks for the audience for tuning in.
Thank you so much for your time, Bob, and I'll see you next week. Ken, was there anything that you wanted to do for house cleaning? And again, thank you for all the work that you do behind the scenes to make this happen.
Yeah. I've got a show tomorrow with I Grow Soil on at 4 p.m. Pacific. So you guys are going to want to check that out tomorrow. Other than that, the regular Andy's on and myself on Wednesday. But Bob, thank you. I know you can't hear me, but thank you.
Thanking you, Bob. That was Ken thanking you.
Thank you, Ken. Had a good time. We'll see you next week.
All right. See you all next week.
Okay. Ciao.
Bye, everybody.